Welcome back, everyone. We're diving into some really cool research today about how our cells are basically like tiny communication networks.
Yeah, it's like they have their own postal service going on, but instead of letters, it's these molecules that control everything, like how they grow and what their jobs are, even when they die.
So we're talking cell signaling here.
Yeah.
But, we've covered that before. What's new and exciting in this research?
Well, the really interesting stuff is how scientists are starting to learn, like, hack this system.
Hack it.
Yeah, like they're figuring out how to manipulate those signals, and it's opening up all kinds of crazy possibilities for new therapies.
Okay, now that sounds promising. So where do we start with this?
So we're going beyond just that basic idea that signals just sort of spread out evenly within a cell. It's actually way more organized than that. We're talking about these little compartments called microdomains where signals are like super localized.
Microdomain. Okay, I'm going to need a visual here. Like, can you paint a picture of what we're talking about?
All right, so imagine a city, right? You've got all these different districts, each one with its own thing going on.
Got it.
Cells are kind of like that. They have these distinct microdomains, each one handling specific signals, even though they're all part of the same cell.
So instead of a citywide announcement, it's like targeted messages being delivered to specific neighborhoods.
Exactly. Much more efficient that way, right?
Definitely.
Yeah.
But how do cells even create these compartments in the first place? What's the infrastructure there?
So that's where these things called A-kinase anchoring proteins come in, or AKPs for short.
AKPs.
Yeah. And they basically act like zip codes, making sure signaling molecules get delivered to the right address within the cell.
AKPs. Okay, I'm writing that down, but hold on. Before we get too deep in the weeds, can you break down what those AKPs actually do? Like, what's their job description?
Sure. Think of them like scaffolding. They hold onto these signaling molecules, keep them organized in specific areas of the cell. One of the key molecules they bind to is protein kinase A. It's a big player in a lot of cellular processes.
Okay, protein kinase A, I think I've heard of that before.
Yeah.
It's an enzyme, right?
Right, and there are actually different types of protein kinase A. Like type 2 enzymes, they're the ones that are really good at binding to AKPs. So they're usually found in specific spots within the cell. Then you've got type 1 enzymes, which are more like free agents floating around the cytoplasm.
Got it. So the type of enzyme kind of dictates where it hangs out in the cell. But how does an AKP actually grab onto a protein kinase, a molecule? Like, what's the connection point?
Right, so proteins have all these different shapes and regions, right?
Right.
Well, protein kinase A has these things called dimerization and docking domains. And AKPs have this special structure called an amphipathic helix that fits right into those domains.
Dimerization and docking domains.
Amphipathic helix.
Okay, I'm trying to picture all of this. Is there maybe a simpler way to explain how this binding works?
How about this? Think of a key fitting into a lock.
Okay, I like that.
The amphipathic helix is like the key, perfectly shaped to interact with those dimerization and docking domains on protein kinase A, which is the lock. And this whole interaction is very specific. It makes sure the right signaling molecules are in the right places.
Okay, it's all about shape and these precise interactions. So basically, the AKPs are like the organizers, making sure those signaling molecules aren't just floating around randomly and that they're in the right spot to actually do their job.
Exactly.
But how do they know where to go in the first place?
That's the cool part. Each AKP actually has this targeting domain.
Targeting domain.
Yeah, and this domain directs it to a specific location within the cell. Like it might bind to other proteins or membranes. It might even have lipid molecules attached that anchor it in place.
So it's like each AKP has its own built-in GPS system.
Pretty much. And this whole intricate system of compartmentalization, it's absolutely essential for cells to function properly. It lets them process all these different signals at the same time without everything going haywire.
I have to admit, this whole microdomain concept is blowing my mind.
Yeah.
It's like cells are way more organized than I ever realized.
Right. It's not just a jumble of stuff in there.
Not at all. It's like this dynamic, bustling city,
Yeah.
all these different neighborhoods working together.
I like that analogy. And speaking of complex systems, let's switch gears for a minute and talk about another fascinating group of molecules.
Okay, hit me.
Thyroid hormone receptors, or TRs.
From AKs to TRs. We are really jumping around the cellular world today. So what are these TRs all about?
Well, they're kind of like master regulators. They're involved in all sorts of processes in vertebrates. You're talking brain development, hearing, bone growth, metabolism. The list goes on.
Wow. Okay, they sound pretty important. What's their MO? How do they actually exert their influence on all those different processes?
They act as transcription factors. Basically, they control which genes get turned on or off. They bind to specific DNA sequences called thyroid hormone response elements, and depending on whether or not this hormone, T3, is present, they can either activate or repress gene expression.
So they're like switches then, flipping genes on or off based on the presence of this hormone.
Exactly. And that whole switching ability, it's crucial for normal development and metabolism. But of course, sometimes things go wrong, and that's when you start seeing diseases like resistance to thyroid hormone
Oh.
or RTH. It's a genetic disorder caused by mutations in the TR gene, and it basically messes up how the body responds to thyroid hormone.
So if the switch is faulty, it can throw off all these vital processes. It just goes to show how important these receptors are.
For sure. And it gets even more interesting because these receptors, they're not just about turning genes on or off. They also have what we call non-genomic activities.
Non-genomic. Okay, now you've got my attention. What does that even mean?
It means they're doing things outside of the nucleus, possibly in the cytoplasm, that don't directly involve gene regulation. We're still figuring out all the details, but it just adds another layer of complexity to these molecules.
So we've got these master regulators controlling gene expression, but they're also involved in all this other stuff we're just starting to understand.
Yep. Cells are busy places, constantly sending and receiving messages.
No kidding. They're like
Yeah.
bustling cities with their own intricate communication networks. Which reminds me, didn't you want to talk about another key player in all this?
Oh, yeah. Let's move over to the nervous system now and talk about glycine in the spinal cord.
Glycine. Wait, isn't that an amino acid? I thought those are the building blocks of proteins.
They are, but glycine also plays a crucial role as a neurotransmitter.
Interesting. So what does it do in the nervous system?
Well, it's the main inhibitory neurotransmitter in the spinal cord. You see, while GABA is the primary inhibitory neurotransmitter in the brain, down in the spinal cord, glycine is the star of the show.
Inhibitory. So it's like a stop sign for nerve signals.
Precisely. Think of it this way. Neurons talk to each other using electrical signals, right?
Right.
And inhibitory neurotransmitters, they act like brakes, preventing those signals from firing.
So it's all about balance, making sure the right signals get through at the right time.
Exactly.
But how does glycine actually do that? Where does it fit into the picture?
Okay, so you've got these things called glycine receptors on the surface of neurons.
Okay.
When glycine binds to its receptor, it causes a change in the electrical charge inside the cell. That makes it less likely to fire an action potential.
So glycine attaches to its receptor and boom, the signal gets blocked. Like a gatekeeper controlling the flow of information.
That's a great way to put it.
But are these receptors just simple on-off switches?
Not quite. They're more like dimmer switches. They have different states.
Dimmer switches. Okay, explain that.
So there's the closed resting state where no glycine is bound, the open state where glycine is bound and the receptor is active, and then this closed desensitized state.
Desensitized. What happens then?
That's when the receptor becomes less responsive if it's been exposed to glycine for too long.
Interesting. So it's not just on or off. There are levels of activity.
Exactly.
But why is that important? What happens if these receptors aren't working right?
Well, that's when you start seeing neurological disorders. For example, you can get mutations in the genes that code for those glycine receptors, and that can lead to something called hyperekaplexia.
Hyperekaplexia. I've never even heard of that.
It's a condition where people have exaggerated startle reflexes. Like if
Right.
they hear a loud noise, they might jump way more than you'd expect and their muscles stiffen up.
Wow. So if the brakes in the nervous system aren't working right, you can have all these involuntary movements.
Right. It's a great example of how crucial it is to understand these signaling pathways, not just for basic science, but also for developing treatments.
Absolutely. And on that note, I think it's time we took a quick break. When we come back, we'll dive into how scientists are actually using their knowledge of these pathways to create new therapies.
It really is amazing how much control these tiny molecules have. And, this whole idea of signaling pathways, it's not just about basic biology anymore. Researchers are using this knowledge to develop some really cool new therapies.
That's what I'm excited to hear about.
Mm.
We've explored all these intricate mechanisms, but how are scientists actually using this information to create treatments? Where's the connection?
Well, you remember those developmental pathways we talked about earlier, right? Like Notch, Hedgehog, and Want.
Yeah, those were the ones involved in cell growth and specialization.
Exactly. And they're essential for normal development, but they can also be kind of hijacked by cancer cells.
Hijacked.
Yeah, like cancer cells can exploit those pathways to fuel their own growth, that uncontrolled growth that's characteristic of cancer.
So it's like they're tapping into these ancient programs, using them for their own nefarious purposes.
Pretty much. And that's where targeted therapies come in. Scientists are developing drugs that specifically block those pathways, basically cutting off the fuel supply for the cancer cells.
Okay, I see the potential here. It's like taking away their cheat codes. But can we break it down a bit further? Start with the Notch pathway. How are researchers targeting that one?
Okay, so the Notch pathway, it's all about communication between cells. It helps them decide what they're going to become, and it controls things like cell division and cell death.
Sounds important.
It is. It's crucial for normal development, but
Hmm.
when things go wrong with Notch signaling, it can lead to all sorts of problems, including cancer.
What kinds of cancer?
Oh, various types. Leukemia, breast cancer, lung cancer, you name it.
Wow, so Notch is kind of a double-edged sword. Essential for normal development, but can also turn against us. So how are scientists disarming it?
Well, one promising approach is using something called gamma secretase inhibitors, GSIs for short.
GSIs.
Yeah, and the gamma secretase, that's an enzyme that's really important in the Notch signaling pathway.
Okay.
So by blocking this enzyme, GSIs can effectively inhibit Notch signaling, essentially putting the brakes on the growth of those cancer cells.
GSIs. They sound like powerful tools. But I'm guessing it's not as simple as just shutting down the entire pathway. There must be some challenges.
You're right, there are definitely challenges. One of the big ones is side effects.
Side effects.
Yeah. GSIs can cause problems, especially in the gut. See, Notch signaling is also important for maintaining the lining of your intestines. So blocking it can lead to gastrointestinal toxicity.
Ah, so it's a balancing act and trying to hit the cancer cells without causing too much damage to healthy tissues.
Exactly. And that's why scientists are constantly trying to improve these therapies. They're looking for ways to deliver GSIs more precisely, maybe targeting them directly to the tumor.
I can see that being a game changer. But let's move on to the next pathway. What about Hedgehog? How does that one fit into the cancer picture?
Right, Hedgehog. Another fun name, right?
Definitely. Doesn't sound very threatening.
I know, but it can be. The Hedgehog pathway, it's another major player in development. It controls things like cell growth and patterning, especially during embryonic development.
So it's like a blueprint for the body.
Yeah, that's a good way to think about it. But sometimes the Hedgehog pathway gets reactivated in adult tissues, and that's when things can go wrong.
Like a program that accidentally gets turned back on.
Exactly. And when that happens, it can lead to the development of certain cancers.
Which ones?
Oh, several types. Basal cell carcinoma, medulloblastoma, pancreatic cancer. Those are some of the big ones.
Okay, so Hedgehog is definitely one to keep an eye on. How are scientists trying to control its activity?
One approach that's been really effective is using these small molecule inhibitors.
Inhibitors.
Yeah. And these inhibitors, they target a protein called smoothened. It's a key component of the Hedgehog pathway.
Smoothened. Another interesting name.
Right. I don't know who comes up with these names. But anyway, by blocking smoothened, these inhibitors can shut down Hedgehog signaling, stopping those cancer cells from growing out of control.
So are there any specific drugs in that category that have actually shown promise in treating cancer?
Definitely. One that's been really successful is a drug called vismodegib. It was actually the first FDA-approved Hedgehog pathway inhibitor, and it's used to treat advanced basal cell carcinoma.
Vismodegib. Okay, I'm making a note of that one. But are there any downsides to using this inhibitor?
Unfortunately, yeah. While it works great for some people, it can also cause some unpleasant side effects.
Like what?
Things like muscle spasms, hair loss, and a weird metallic taste in your mouth.
Ooh, that doesn't sound fun. It seems like there's always a trade-off with these powerful therapies.
There often is, unfortunately. But that's why research is so important, constantly trying to find ways to improve these treatments and minimize the risks.
For sure. And speaking of ongoing research, let's move on to our final pathway. What about want? What makes that one unique, and how is it being targeted for therapeutic purposes?
Okay, so want, it's another one of those complex signaling cascades that's involved in a ton of different processes: cell proliferation, differentiation, migration.
Fizzy little pathway.
Right. It's essential for embryonic development, tissue regeneration, maintaining stem cell populations. It's all over the place.
Wow, it sounds like a real multitasker. But how does it go wrong in cancer? How do those normal functions get twisted into something harmful?
Well, if the want pathway gets dysregulated, through mutations or abnormal activation, it can lead to uncontrolled cell growth. And that, of course, can lead to tumor formation.
Okay, so want is another one of those essential pathways that can turn against us.
Exactly. It's been implicated in a bunch of different cancers: colon cancer, breast cancer, melanoma, you name it.
So how are scientists trying to target it?
There are a few different approaches they're looking at. Some researchers are trying to inhibit the production of want ligands. Those are the molecules that activate the pathway. Others are developing antibodies or small molecules that block want receptors.
So cutting off the communication at different points,
Ah.
either prevent the message from being sent or block the receiver from getting it.
That's the idea.
Pretty clever. And are any of those approaches actually showing promise in clinical trials?
Yeah, there are some exciting developments. For example, there's this monoclonal antibody called vantictumab.
It's too mad.
It targets want receptors, and it's actually shown some encouraging results in early-stage clinical trials for pancreatic cancer and breast cancer.
Interesting. But I'm sure there are challenges here too, right? What are the downsides of targeting the want pathway?
Well, one concern is that it could have off-target effects. You see, the want pathway is involved in so many important processes that messing with it could have unintended consequences. Like, it's really important for tissue regeneration and maintaining stem cell populations.
Right. So we need to be careful not to disrupt those processes.
Exactly. And that's why scientists are focusing on developing inhibitors that are very specific. They only target the parts of the pathway that are involved in cancer. And of course, they're doing lots of testing to make sure these treatments are safe.
Makes sense. It's incredible how much progress has been made in understanding these pathways and using that knowledge to develop new cancer therapies. But I'm also curious about the other side of the coin. We've talked a lot about blocking these pathways, but can we also harness them to promote healing and regeneration? Now, that's a really cool thought. It's like we've been focusing on the dark side of these pathways, but what about the good stuff? Like, can we actually use them to help the body heal and repair itself instead of just blocking them?
Well, that's what's so exciting about this whole field of regenerative medicine. And the short answer is yes. We're making huge strides in using these very same pathways to actually encourage tissue repair and regeneration.
I knew it. So what's the state of the art here? Are we talking about stuff that's way off in the future, or are there already therapies being used?
You might be surprised to hear that some of these therapies are already being used. Like, one area where regenerative medicine is showing a lot of promise is in treating cartilage damage, especially in joints.
So instead of just going straight to joint replacements, we could actually stimulate the growth of new cartilage cells.
Exactly. It could really change the game for people dealing with joint pain and limited mobility.
That would be incredible. So how does it work? How do you actually get the body to regrow its own cartilage?
Well, there are a few different techniques. One that's being used is platelet-rich plasma injections.
Platelet-rich plasma.
Yeah, it's basically where they take a sample of your blood and concentrate the growth factors from the platelets, and then they inject it right into the damaged joint.
So you're basically using your own body's healing power, just giving it a little boost.
That's the idea. Another technique is called autologous chondrocyte implantation.
Autologous chondrocyte implantation. Okay, that one's a mouthful.
It is a bit of a tongue twister. But basically, they take a small biopsy of your cartilage, grow more cartilage cells in the lab, and then implant those cells back into the damaged joint.
So it's like giving your body a personalized repair crew.
Pretty much.
That's amazing.
Yeah.
But cartilage is just one type of tissue. Are there other regenerative feats that scientists are working on?
Oh, absolutely. Like regenerating heart muscle after a heart attack. That's another big one.
Wow, to actually mend a broken heart. I mean, not just metaphorically, but physically.
I know. It sounds pretty incredible, right?
Yeah.
Heart attacks can do a lot of damage, leaving behind scar tissue that affects how well the heart functions. But what if we could stimulate the growth of new heart muscle cells and actually reverse that damage?
Now, that would be a game changer. Are we getting close? Are there any therapies that are looking promising for that?
One of the most exciting approaches uses something called induced pluripotent stem cells, or iPSCs.
iPSCs?
Yeah. These are basically adult cells that have been reprogrammed back into a state where they can become all sorts of different cell types, including heart muscle cells.
Wait, so you're telling me we can take, say, a skin cell and turn it into a heart cell?
That's exactly what I'm saying. And by exposing these iPSCs to the right signals and growth factors, the same ones we've been talking about, scientists can actually guide them to become heart muscle cells and even encourage them to integrate with the existing heart tissue.
That's mind-blowing. It's like giving those cells a whole new set of instructions, a new purpose.
It really is. And heart muscle and cartilage are just two examples. Scientists are working on regenerating all kinds of tissues: spinal cord tissue after an injury, damaged nerves, even limbs. There's even research going on in restoring vision loss.
It feels like we're on the verge of a whole new era in medicine.
I think we are. We're going from just managing symptoms to actually repairing the damage at the source.
It's incredible.
Mhm.
But none of this would be possible without the incredible work of scientists from all different fields.
Right. It's a true team effort.
Right.
You've got cell biologists, geneticists, engineers, material scientists. It's amazing to see what they can accomplish when they put their heads together.
So as we wrap up this deep dive into the fascinating world of cellular signaling, what's the takeaway message for our listeners?
Well, I think the key message is that we're living in an era of incredible scientific discovery. We're unlocking the secrets of these ancient pathways that control life itself, and we're learning how to use that knowledge to both fight disease and promote healing.
It's pretty amazing, isn't it? It makes you realize just how complex and resilient the human body is.
It really does. And who knows, maybe one of our listeners will be inspired by all of this and go on to make their own contributions to this exciting field.
That would be fantastic. And on that note, we'll leave you with this thought. The future of medicine is brighter than ever, and it's all thanks to the amazing work of scientists who are constantly pushing the boundaries of what's possible. Stay curious.